I would like to introduce our two prestigious speakers from Matthias Mann's lab, Özge and Lisa. Thank you for joining from Germany. So Özge is a very old friend, actually my very special student from my time from Koç University. She's a graduate of Koç University. And after her bachelor degree, she continues master's at the Koç University in my laboratory. She did quite a bit of proteomics projects and published 3 nice proteomics articles, and then moved to Matthias Mann's lab for her PhD, and there she also did very amazing job. She's currently a postdoc there, but I know she will be moving soon to the US. So she's in the Max Planck Institute for Biochemistry now, and she works in the field of mass spectrometry-based proteomics, and her research is mainly in the development of sensitive and quantitative proteomic strategies for studying phospho and ubiquitin signaling in health and disease. And she also did quite a bit of clinical proteomics, as far as I know. And our second speaker, Lisa Schweizer, is also from Matthias Mann's lab. She did her master and bachelor degrees in biochemistry at Technical University of Munich. And in 1918, she groups Mann's group in Max Planck Institute, and continued her PhDs. And she's doing MS-based proteomic characterization of 4 million fixed paraffin-embedded tissues. And she's quite a bit of working with the clinical pathologies, and we also congratulate her for her award in the society, German Society of Pathology in 2020. And her research focuses on the resolution of cellular heterogeneity in cancer and other complex human disease using MS-based proteomic approaches. Today, you will be both presenting a very hot topic, and I know Mann's group has published quite a bit of nice studies on this, which is the proteomic analysis of the SARS COVID virus, and we are looking forward to your presentation. I think, Özge, you will be presenting first, right?
Yes, I will be going first.
Share the screen. Okay, thank you.
Do that. Can you see it?
Yes, we see it very nice. Yes.
Okay, perfect. So, yeah, thank you so much for the invitation and this nice introduction. As a Turkish scientist who also met proteomics and got the first proteomics training in Turkey, I am delighted to be the first one presenting at this platform. It's always wonderful to see my mentors and amazing peers from Turkey, and I am extra happy today because I do this with Lisa, who is actually a wonderful colleague and brilliant scientist from Matthias's group. And today what we want to show you is that the complexity actually many of us face every day. It doesn't matter whether you're doing wet lab or bioinformatics. As proteomics community, we are challenged by this remarkable complexity of the proteome. And we all try to resolve it, right? Maybe in different ways, but with a common goal, which is to better understand biological mechanisms. So for instance, what you see here is the mess created by SARS-CoV and SARS-CoV-2 infections, and more than 1,000 interactions between host proteins and viral proteins. And the point that I want to make here is that, unfortunately, biology is never easy, and SARS-CoV-2 biology too. And actually, within the last few decades, not only us, but the entire scientific community started to appreciate that human proteome is vastly more complex than the human genome. And there are mechanisms such as genomic recombination or alternative splicing that allow cells to generate different mRNA transcripts from a single gene. And even though the neuronal and the muscle cells in your bodies have the same set of genes in their nucleus, the functional proteome makes them different. And one way to actually increase the functional diversity and complexity of proteome is facilitated by post-translational modifications, which regulates cellular ability to adapt quickly to many changes happening in our internal and external environments. And of course, mass spectrometry-based proteomics enables us to investigate nearly complete proteomes today. And this technology has also evolved into the method of choice for studying PTMs because of its unbiased nature and ability to locate PTMs with a single amino acid resolution. So given the central role of proteins in biochemical pathways, they are likely the most ubiquitously affected in disease response and recovery. And MS-based proteomics holds a special promise for the unbiased and system-wide analysis of clinical phenotypes. And from a broader perspective, Lisa and I have the similar vision, which is to address clinical needs by either translating or developing reproducible, sensitive, and high throughput MS strategies so we can bring proteomics into clinic to improve individual health. So using cell lines, biofluids, biobank tissues, we actually aim to systematically profile health and disease states to achieve a comprehensive understanding of disease mechanism, mostly at the protein level, as Lisa will tell us today, but also PTM levels. This is going to be the part that I will be showing. So PTMs are great because you add a little modification to a protein, and in this way regulates its activity, localization, and interaction with other cellular molecules. So they are kind of vital control mechanisms, right, governing almost all major aspects of cellular life, including cell cycle and protein degradation. So, and we also know that aberrant PTMs are associated with a variety of human diseases, including neurodegeneration and cancer. So there are about 200 different PTMs identified so far, but today in this talk, I will be focusing on arguably the most prevalent ones, phosphorylation and ubiquitination, which are in fact very different. While phosphorylation is a remarkably simple, small chemical modification, ubiquitination means the covalent attachment of another small protein called ubiquitin. So as you all know, phosphorylation, this reaction is catalyzed by kinases, and the removal of the phosphate group is catalyzed by phosphatases. And we know that over 500 kinases and 100 phosphatases are found in the human genome. What is amazing is if you can analyze 14,000 proteins in a cell lysate, you may find as many as 1 million phosphorylated sites. This really tells us that the protein phosphorylation is widespread and modifies the activity of many, many, many proteins in the cell. And ubiquitin fold is also really nice because it's a versatile model, and it can comprise structurally conserved protein family, which can regulate virtually all processes in eukaryotic cells. And this process is relatively a bit more complicated than phosphorylation because it requires a cascade involving multiple enzymes, but all these enzymes are highly important to control the efficiency of the ubiquitination reaction and to confer substrate specificity. And so far, 2 E1s, a limited number of E2s, and hundreds of E3s, and more than 100 deubiquitinating enzymes have been identified in humans. So what I like here is this. So one kinase or one E3 ligase can target hundreds of potential substrate, enabling sort of signal diffusion across many signaling networks, typically inducing various unique biological responses. And this complexity of signaling networks has recently been more acknowledged thanks to the MS-based proteomics, right? But for a really long time, the application of MS to PTM analysis has brought the need for prefractionation and enrichment to reach the depth required to cover key signaling molecules. This was mainly due to the fact that only a small fraction of peptides in complex mixture contains modified peptides, And those peptides usually present at substoichiometric levels, meaning that they are less abundant than their non-modified counterparts. And therefore, these workflows usually mean that you need a lot of protein material, making them unapplicable for clinical samples. However, it turned out that DIA, data-independent acquisition-based workflows, is also very well suited to large-scale analysis of not only proteins, but also PTMs. So I guess it's also worth mentioning at this point, maybe very briefly. So the popular enrichment technique for phosphorylated peptides is affinity-based chromatography. That's also the one we use, which takes advantage of positively charged metal ions presented for the interaction with negatively charged phosphate groups. But for ubiquitin, we use anti-digalide antibodies, and those antibodies interact specifically with the ubiquitin-derived glycolide, digalide remnant on ubiquitinated proteins after trypsinization. So data-dependent acquisition, DDA and DIA, are the common strategies that we use in shotgun proteomics today. DIA is just relatively newer approach compared to DDA, but the traditional DDA takes only a selection of peptide signals forward for fragmentation, and then matches them to a predefined database. But in contrast, in DIA, the quadrupole continuously cycles across the entire mass range while selecting relatively large M over Z values. And therefore, we can say that it's unbiased and better technique for discovery proteomics. And now the studies also show us that it can identify more peptide precursors than DDA and shows higher precision and better reproducibility with fewer missing values. However, DIA generates a very complex MSMS spectra, but nowadays the modern software can easily deconvolute, the spectra to identify multiple peptides from a single scan, usually by comparison to a previously acquired peptide or spectral library, however you call it, but increasingly also without. So in a team effort with Finn Hansen and Maria Tanzer from our group, we have been investigating the power of DIA for large-scale analysis of phosphorylation and ubiquitination events in a single run analysis format. So we have started this roughly 4 years ago, while it was still quite new, but today there are several other studies actually demonstrating superior performance of DIA for, PTM, for the analysis of PTM enriched samples, mostly phospho-enriched samples. But as I said, so the need of relatively larger sample amounts and extensive fractionation on top of enrichment actually comes at the expense of throughput, robustness, and quantitative accuracy. But DIA can now overcome most of these problems and substantially simplify workflows. So what we do here is we build a comprehensive spectral libraries for efficient extraction of modified peptides in single-shot DIA analysis. So we basically take the conventional workflow to build our libraries. But when it comes to actual samples, we can actually reduce the amount we need, for the actual study samples by almost by factor 10. So Finn's study on DIA-based analysis of ubiquitinomes is a beautiful and prominent example of what I've been trying to explain. So for instance, here, to obtain a very good spectral library, which we can always use in DIA-based ubiquitin studies, he separated peptides by basic reversed phase chromatography and isolated the fractions containing the highly abundant K48 link ubiquitin chain drive digalide peptide and processed those fractions separately. And this actually helped him to reduce the excess amounts of K48 peptides in individual pools, because it can actually compete for antibody binding during enrichment. And also this giant peak would also interfere with the detection of collating peptides. And then from 2 human cell lines, we obtained around 90,000 digalide sites corresponding to more than 94,000 digalide peptides. Half was detected in at least 2 libraries, 2 cell lines. And to best of my knowledge, so this represents the deepest diagonal proteome data to date. And this is now available actually for you to explore in the Phosphosite Plus database. And then by having repeated measurements of the same diagonal enriched sample using both DIA and the DDA methods, we actually showed that DIA approach almost doubled the depth of ubiquitinum analysis in single runs and substantially increased the reproducibility. So how we judge this, it's basically based on the number of distinct diagonal peptides with coefficient of variation, CVs below 20 and 50%. And we always do this comparison, to an optimized DDA method. Is that all? But of course not, right? Over time, our library generation strategy has also evolved a little bit with advances in the technology, and especially after we switched to Thermo's one of the new platforms called Explorers. So what is nice about this platform that it comes with FAMES interface, which involves separation of ions when entering mass spectrometer based on their inherent differences in their mobility in high and low electric fields. And the great thing is that this technique can be applied in atmospheric pressure and room temperature. And we utilize it to perform sort of a gas-phase fractionation on the fly, and for this, we basically take an aliquot, from actual study samples and we pool them. And then in this way, we generate our DI libraries. In the interest of time, I kind of ignore one important step here because we haven't really published it yet. But at least I can tell you that we have also automated almost every steps of our PTM sample preparation workflow. So those tailored upfront steps, together with this smart way of generating DIA libraries, resulted in a great pipeline. And we can now use this pipeline to study PTMs at large scale and further reduce the sample requirements by almost factor 10 for ubiquitination and more than 5 for phosphorylation, so which is actually amazing. But what I want to show you is we have utilized very early versions of those sensitive and scalable pipelines to study host signaling processes affected by the SARS-CoV and SARS-CoV-2 at the levels of protein, phosphorylation, and ubiquitination. And for this project, we collaborated with Andreas Pickermeyer from TEM. And I must say that I still feel blessed to be included in this project. And it was such a nice journey to take with all amazing scientists from his team. And together we succeeded in obtaining a large molecular picture of infection process in lung cells. So we have a beautiful website for you. It's just waiting for you to go and type in the name of your favorite protein to see if and how it is affected during infections by coronaviruses. And of course, our study is not the only one utilizing proteomics, right? Or proteomics approaches to study SARS-CoV-2 biology. Now there are like many stellar publications out there which came out around the same time of ours or after that. But what I want to highlight here is that in one study, we did a remarkable job, in my opinion. We profiled the impact of viral infection on mRNA transcription, protein abundance, ubiquitination, and phosphorylation in a time-resolved manner by quantifying thousands of entities, biological entities. And that at the end allowed us to identify regulatory circuits that are affected by these viruses. And on top of it, we studied the interactome, as well as activities and the effects of individual viral proteins in a single study, right? And this actually gave us a huge amount of information. And very interestingly, we have shown that viral proteins are phosphorylated and ubiquitinated when they're produced in the cell. And we even suggested the enzymes that may catalyze these events. And of course, at the end, as I said, we have had a huge information about the dynamic modulation of multiple host signaling pathways And in the interest of time, among many interesting findings, I just want to show you one example because, you know, just to demonstrate you what you can really do with such an amazing data set. So actually here, enriching functional connections with multiple omics data set pointed towards the potential mechanisms of autophagy regulation by viral proteins, namely ORF3 and NSP6. And ORF3 is actually an important player. So we observed a significant inhibition of autophagic flux by ORF3, leading to accumulation of autophagy receptors, which we can judge by looking at their, how their abundances are changing over time. And this phenomena was actually correct in both viral overexpression and time course infection data. And this inhibition may be explained by the interaction of ORF3 with the Hobbs complex, because this complex is essential for autophagosome-lysosome fusion, as well as the differential phosphorylations or ubiquitinations of key components in this pathway. Of course, you may ask how such examples can help to fight COVID-19, right? So to this end, Andreas's team actually searched for the drugs targeting unique and common molecular mechanisms, so-called hotspots in cells, modulated by these viruses. And after identifying around 70 drugs in combination with a functional assay, testing the effects of these drugs on viral replication, they actually suggested and identified several promising antiviral treatments. And we have included these results also in the manuscript. I really hope that the sensitive and robust DI-based PTM analysis workflows are also of interest to you. And they will be used in future to uncover PTM events regulated in various cellular processes in health and disease. So I think with that, I will just give the stage over to Lisa. Should I stop sharing?
Okay, so I will share my screen now. And I hope you see my presentation now.
Yes, yeah.
Really good. So, okay. Then, after this great insight into the mechanism of infection and maybe also progression of SARS-CoV-2 in lung cells, I would now like to maybe ask you to change a bit the perspective on a human cell, and ask you to look at this picture of the salivary gland that actually shows that cells in the clinical context or in the body are embedded in individual architectures of tissue and thereby surrounded by other cells or by extracellular matrix. And if we now start from this really embedded tissue of these embedded cells here, this is of course of high importance for clinical research to find what's in the surrounding of the cell and what do cells really do in the environment of the tissue. So in the clinical context, tissue is often derived or mostly derived from biopsies, but also from more invasive surgeries from patients, and then stored as FFPE tissue. And FFPE stands for formalin fixed paraffin embedded tissue, which means that the proteins are fixed by formalin crosslinks in the proteins, and then the whole tissue is embedded into a paraffin matrix to stabilize it. And due to the stabilization, the tissue or the stabilization allows the tissue to be cut into small slices for follow-up immunohistochemistry, for example, but also allows stability to store this tissue at room temperature without the requirement of expensive freezers or huge space to store the tissue. And actually also the proteome is stable for decades in the state. Next to this advantage of the storage of FFPE tissue, we also for our clinical research have huge advantage of this tissue because these tissues are often stored in archival databases and associated to clinical metadata such as age, as treatment, as other drugs, which is associated to this tissue information. And given these both advantages, to date, approximately 500 million FFPE specimens are stored only for cancer worldwide, which is a representation, a reflection of how popular and standardized this FFPE format to store tissue is actually. And for now, as researchers, as you see here in the bottom of the picture, now for mass spectrometry-based proteomics, one of the key aspects to analyze clinical tissue is a high throughput and comprehensive workflow to analyze these tissues. And I only want to give you 2 examples of various possibilities to prepare this tissue, which differ by the requirement of the upfront removal of paraffin from the main sample. So in this first strategy, as you see here on the slide, we first manually, for each single sample specimen, deparaffinize tissue as it's done in the clinic with xylol and a row of ethanol dilutions to only get the tissue without paraffin in the end on a microscopy slide. Subsequently, either by laser capture microdissection or also by macroscopic dissection, tissue can be collected in a 96-well format. And actually in this 96-well format, we can do or perform the lysis, the decross-linking of the formalin bridges, as well as the proteomic or tryptic digest in a one-pot, single-pot reaction to then yield peptides that can be injected into mass spectrometer. And this whole outline in the single 96-well format actually allows us to perform single-pot reactions and actually also to automate the sample preparation, which perfectly meets the requirement of clinical sample preparation. For an alternative strategy, we aimed to prepare or to provide a more universal sample preparation strategy without the requirement of upfront deparaffinization. So also for this approach, FFPE sample, here shown as an example of adenoma tissue, is collected in a 96-well format, and the paraffin and the proteins are separated from each other by alternative steps of heating to 90 degree or 95 degree and focused sonification on the tissue, leading to a milky whitish color of the sample, as you see here in step 1. Subsequently, the proteins have to be extracted from this paraffin solution, which is done by a protein capture on magnetic beads, as you see here in step 2, which allows then to perform washing steps on these beads by a magnet that you apply on these beads, using 100% isopropanol, which is far, far less toxic than the xylol that we used before, and heating up to 50 degrees. During several wash steps, namely 3 in this case, we are then able to remove the paraffin from the first step, as you see here on the left-hand side in step 3, towards the third step on the right-hand side, which yields in a really nice and clear solution, which is ready to go for the injection into the mass spectrometer. So in summary, this protocol here provides an opportunity for universal sample preparation, if a tissue is paraffinized or not paraffinized, to pool all these tissues and to process them in parallel in a high throughput format. With these workflows in hand, actually now with the first cases of COVID-19 in spring of last year, the necessity and also the urgent requirement for deep biological information of COVID-19 and also for high throughput format of sample processing got even more important throughout the last 1.5 years. So in spring 2020, our colleagues from the University Hospital in Augsburg performed a post-mortem examination of a case series of patients with fatal COVID-19 and were among the first groups to describe the serial autopsy of all different organs of the body, as well as the histologies of COVID-19, which is shown on the right-hand side here for the lungs as organizing an end-stage diffuse alveolar damage of the lungs, as well as some other cellular specifications, such as hyaline membranes, which are shown here by the black arrows on the left-hand side, as well as metaplasia in the lung tissue. And while we now see here this histological impression of COVID-19, for us as proteomic people, of course, we are now interested if actually this histology and this phenotype is also reflected in close correlation to the actual molecular phenotype by the proteome. Driven by this question and in close collaboration to our partners in Augsburg, we then set up a study which intended to be across 10 different organs of the human body, including the lungs, but also other tissues such as the lymph nodes and the brain, and for example, also the heart or the spleen. Based on these tissues, also here we collect these tissues into 96 well plate and applied our readily applicable and universal sample preparation workflow using the magnetic beads, and further streamlined our method using a standardized liquid chromatography that was initially also designed to be applicable in the clinics. Based on this whole streamlined workflow, we identified more than 7,300 proteins across all organs using, as Özge explained before, a DRA data acquisition mode, and even more important, the processed and acquired data from more than 350 samples within only 26 days of measurement time. Now, focusing on these more than 7,300 proteins and comparing significant proteins in the comparison between the COVID state and healthy or non-pathological control states, we identified, as we would expect, the biggest changes, the most significant changes in the lungs, as you see here on the left-hand side, but surprisingly also in a lot of other tissues like lymph nodes and the blood vessels, so the lymph vessel system, but also other organs like the liver or even the brain, even to less extent. When going a bit more into details of these differentially expressed proteins, we identified 3 major processes across all organs, which is the fibrin and platelet aggregation, the complement system, and the interferon system, which all of them show a clear upregulation in the lungs, but also in the other organs, as you see outside of these red boxes. Now, further focusing on the lungs as major entry point of the virus and also major place of the most symptoms of COVID-19, we subsequently performed a protein-protein correlation analysis of significantly upregulated proteins and downregulated proteins in the lungs, and interestingly identified the major hallmark of COVID-19, which is inflammation, in 2 separate clusters of this analysis. On the one hand, if we focus on cluster A, we see upregulation, but also a co-expression of immunoglobulins in COVID-19 in comparison to other lung controls that we included in our study. On the other hand, focusing on cluster B, we even more strongly observe a high synchronization and upregulation of the interferon answer, not only in the COVID-19 samples here shown on the left-hand side, but also in similar pathologies such as influenza here shown in yellow. With these hallmarks of COVID-19 as a proof of principle and this really huge synchronization of interferon proteins in hand, we then added a level on the proteome analysis. And I would now like to go back to this nice explanation of Özge in the beginning to add a layer to the phosphoproteome analysis. For this purpose, we enriched our proteins using iron anti-A enrichment chromatography on an automated system by Agilent Technologies, as you see here on the right-hand side of the depiction, and actually identified a plethora of phosphorylation sites that were differentially regulated in COVID-19. If we only focus on 2 of them, we see a clear upregulation of proteins associated to mRNA splicing in COVID-19 here on the left-hand side, as well as a clear downregulation of proteins associated to Rho GTPase signaling on the right-hand side of this heat map. Among those, we also find proteins like cycles, just as the RAC1 GTPase cycle, and other very interesting terms that very nicely at some points reproduce the cell or the results that Özge and Andreas Bichemeyer in their study identified as showed before. And now with the proteome and the phosphoproteome in hands in the background, you may also ask, yeah, this is really nice, but how about actually the virus itself? Because also the virus has proteins, of course, and do we actually identify this virus as well? And the answer to this is clearly yes, we do. In some patients, we untargeted identify on-the-fly peptides from the membrane protein, but also from the nucleoprotein in stages either of acute or organizing regions in the lung samples of COVID-19. these results now show not only that we are able to identify peptides in a really unbiased manner, in contrast to, for example, the PCR test, where we really targeted look for the RNA or DNA in the end of the virus, but we here really find on-the-fly identification of SARS-CoV-2 peptides in our whole proteome analysis, which also shows that we actually achieved in the end, using our high throughput and comprehensive analysis and workflow of COVID-19 samples, an in-depth proteomic insight into our samples. And with this summary here, I would like to go once more back to the salivary gland that I presented to you in the beginning, because where we started here, we actually described, okay, now the cells are integrated into the architecture of tissues. And this is also what we're using now in this bulk proteomics approach when really looking on the overall tissue in patients. But in fact, if I now may ask you to again change your perspective and having a look on a single cell and a plethora of single cell in the whole tissue, actually the heterogeneity of cells is sometimes a bigger or diverse than we would expect. So with this image now in mind, of course, normally in bulk proteomics, this cellular heterogeneity is neglected. To resolve now the heterogeneity and the complexity of these tissues, our colleagues in Copenhagen, Fabian Kosch and Andreas Mund, developed a novel technique to resolve this heterogeneity. Termed deep visual proteomics, this method, as before, is based on paraffin or FFPE tissue, which is derived from patients, and which in the first place is stained by immunohistochemistry for cellular boundaries or for specific morphological aspects, and is then recorded using highly multiplexed microscopy to achieve high content images. Based on these high content images, our AI algorithm using deep learning will segment and recognize single cells in the tissue and also classify these cells according to different labels or actually different properties, and export these different classes to a laser capture microdissection microscope, which will then get kind of a template of the artificial intelligence. Based on this template, our laser capture microdissection microscope is then extracting single cells in tissue based on this previous classification and is collecting single cell populations for the follow-up ultra-high sensitive mass spectrometry analysis. Subsequent to this, and going back actually to the spatial resolution, which is preserved in this approach by the artificial intelligence, we are then able to analyze these results in, for example, clinical knowledge graph databases, but also, of course, in close correlation and collaboration to our clinical collaborators. And to just give you an example on how this could look like is one of the recordings that we did actually in our microscope in Munich, which is borderline or low-grade ovarian cancer, shown in a 63x magnification. And as you see here, we see a bright field image of the tissue where you see the cell boundaries approximately by a bit lighter lines, but also the template in red that the artificial intelligence is putting on top of the tissue, computationally, of course, and which guides the laser to extract the single cells and then to collect these single cells by a final pulse into the well, which can then be processed afterwards. And just to give you an impression how this could look like in our salivary gland example, we now go back from salivary gland to carcinoma. And in a very simple case, we are now actually able, just by AI segmentation, to differentiate between normal acinar cells in this gland and actinic cell carcinoma cells, which are shown here in purple. And by comparing these two cell populations, actually, we already see in the Pearson correlation of the proteomes that the correlation is completely different for both of the different cell types, but correlates, of course, within specific replicates, as is shown here on the right-hand side. Now, with this thought and the idea of deep visual proteomics in mind, I would now like to come back to our COVID-19 example and give you a perspective on what could be done in the future using this method. So let's start again with the immunostaining of SARS-CoV-2, which is actually possible by either RNA or also protein-based approaches, and can actually highlight cells that are infected by SARS-CoV-2 and undergo active proliferation of the virus. And as you see here on the left-hand side in figure A, this spread of the cells that are actually infected by the virus is quite diffuse and is not covering the whole lung tissue. So if you imagine you will now compare the bulk proteomic analysis and the deep visual proteomic analysis, we would have a huge advantage to only extract these cells that are in current proliferation of SARS-CoV-2 versus the cells that are not yet maybe infected or have been infected, to actually identify mechanisms that contribute to the proliferation and therefore also to find drug targets that might inhibit this proliferation. And to again summarize this again with this possibility, we could actually identify on only single cell population relevant mechanism of the disease. And with this, I now want to come back to our summary slide and actually add to the first part of our presentation that next to cell lines, we have actually also developed universal sample preparation strategies for formalin-fixed and paraffin-embedded tissue that is now readily applicable for the clinic and can be automated to have a streamlined workflow in the clinic as well. Additionally, our colleagues from Copenhagen and our group currently is promoting or developing the deep visual proteomics technology to resolve the heterogeneity of cells within the tissue, to actually add a layer on top of the old analysis or the recent analysis to identify cells in the actual structure of the tissue. And with this, as we both have showed you today, actually our mass spectrometry-based proteomic workflow is able to answer or to identify critical biomedical questions in the context of COVID-19, not only in cell lines, but also in FFPE tissue. And with the summary, I can only in the end acknowledge our whole group, and first of all, of course, Professor Dr. Matthias Mann, and for the great opportunity to work in this absolutely great research environment at the Max Planck Institute of Biochemistry in Munich, as well as Finn and Maria again, who helped a lot in Özge's study for the SARS-CoV-2 project, and as well as Max, Sophia, and Andreas, who helped majorly in my study, in addition to Özge, of course. And with these images of our maybe nice insights in our social lives of the lab, I would like also to thank you for your attention.
Thank you both Lisa and Özge for this terrific talk. Very nice. I congratulate both of you for having a lot of impact onto the SARS-CoV proteomics. I think this is very nice. So I would like to ask the audience if there are any questions. So you can type your question, or you can just simply unmute and then directly ask the question. As we mentioned this, we try to make this as unofficial as possible. So please ask if you have any questions and don't hesitate. So maybe I can start with the first question. Like, I was very amazed by the DIA for the PTM analysis, and so this is very nice. And like, I wonder how feasible it is, right? So for even phosphorylation, generating a library should be quite a bit of work. And you mentioned you have this fancy mass spec for the gas phase fractionation, so fragmentation. So how crucial is this? Like, can you generate a library with a random machinery for the glycosylation or ubiquitination, like for either PTMs to do DIA?
I mean, yeah, we see that the library generation takes a lot of time and effort still, like if you want to go really deep, even in the single one analysis format for PTMs. But I must say that slowly we are actually switching to direct DIA, which does not require the generation any peptide or spectral libraries. So we can still get a lot of, a good coverage and still quantify easily 10,000, even in some cases like 20,000 modified peptides in a single shot which is actually quite amazing. So with this, the modern software, I don't know if I should name them, but they can actually do it. So yeah, so the data analysis is the software part is actually advancing pretty quickly. So
what software? I think you can say the name of the software.
I guess you also use Spectronaut, so we use Spectronaut extensively. But if you want to go for like a free softwares, now the MaxDI is also available. So yeah, basically the MaxQuant for DI. And Diane is also widely used at the moment. So we also use it heavily in the department. We really like it. So you can also now use Diane for both phosphorylation and ubiquitination. Okay. So yes, but of course, I mean, even if you don't have this, the FAMES interface, I mean, it's also kind of like a fractionation, right? But it's just a gas-phase fractionation. Just you do it on fly while just, analyzing the samples. Even if you don't have it, sometimes
it's the fractionation. Actually, it's not the fragmentation. It's the
no, it's a fractionation. It's a gas-phase fractionation.
Okay, yeah. That's what I heard, but then I corrected myself because like fractionation and gas phase.
No, it's fractionation. Yeah. So if you have like a big project, right, and you want to do PTM analysis, DI-based PTM analysis, sometimes it's actually worth generating such big libraries because you can use it over and over, right, for multiple projects. So you can have one, so it's like such as in Finn study. So we'll build one really good library and we have used it for multiple applications. And this is what we show in the manuscript. So you can even just take those, right, and also adapt the mass spec methods and then directly use them. So we even like most of the time provide the spectral libraries, like as a form of, the software output. They're just like ready to go, basically. You can download and then, upload in the software and then just use it.
I see. Cool. Very nice. So I have another question, if I have time, like a biological question, both Lisa and Özge. So Lisa showed that, like, when you were looking at the lung tissues, the number of proteins seems to be drastically dropped for lung, but not the others. I wonder, are there any extensive proteolysis going on? Did you also see it in the cell lines, like more ubiquitination or something? Why the lung has such an effect in the protein number, but not the other tissues?
Yes, if I may start. So the numbers that we show were these significantly differentially regulated proteins. And so for sure, there's more going on in the lungs than in the other tissues. And actually, the proteolysis is a really interesting aspect. So we have not actively observed it. So if we compare the only identification of proteins in the lungs and in the other tissues, of course, it's always a bit tissue specific sometimes. So for example, in the heart, you usually identify less proteins than in the lungs or lymph nodes, for example. But for a majority of tissues, the protein identification is quite stable. So actually we could guess to exclude, or the majority actually of proteolysis here, but something else is actually happening on the phosphorylation side. So maybe Özge wants to comment on this.
Yeah, exactly. So what can I say? So what we frequently observe that if you add an extra step in your workflow, if you're doing like some PTM enrichment or isolation of specific organelles or something like that. So the complexity, if the tissue is really highly complex, you can sometimes quantify less modified peptides or less number of proteins at the end. This is usually quite stable. If you're just doing, digestion and the global proteome analysis, you don't really see that effect. But that is mainly limited by the, the sensitivity of the, your methods and stuff. But if you're adding additional enrichments in your workflow, you can see that those numbers are highly affected by the complexity of tissues. So for instance, brain, sometimes it's not really easy to get a good coverage because it's highly complex and so on. I don't think that lung is actually one of them. So, but in that case, actually, this was like it was a quite new application for us also too, right? So, because this was like the first time we were doing phosphor enrichment from such a special, tissues. And we did not really get, a super deep coverage, to be honest. I mean, at least like it was like less compared to what we usually get in cell lines, but still it was enough to cover like many key components, you know, or like pathways, components of pathways in lung cells. So it was still okay, in my opinion.
Cool. Yeah, nice. Any other question from the audience?
Again, it was a great talk. Thank you very much, Özge and Lisa. So I have a quick question about the cell line data. Actually, it's a cancer cell line, right?
The one we use, yes.
Yes.
A54.
Yeah. Do you think that the oncogenic pathways dominate the results? And do you have any analysis on that?
Of course, yes. I mean, this is just, you know, this is how it is. So unfortunately, when you work with like, you know, the cancer cell lines, and most of the time, you know, for instance, like there are some pathways that you always see, right? Because they're active and they are most of the time dominating your analysis. But just to avoid that, we try to go like, you know, as deep as possible in this case and try to cover, you know, many pathways, not only the ones which are kind of expected to be, you know, dominating the entire analysis pipeline, but just also the ones which could be like, you know, important for COVID-19. So for instance, the TGF-beta pathway, so which may also play a role in fibrosis, which we were able to cover like the, you know, the major components of that pathway and see like, you know, how they are regulated in a time resolved manner. So yes, of course, but this is like a general problem of, you know, using, unfortunately, cancer cell lines, yeah, in such applications. But I hope that now, I mean, for instance, with ROHO GTPases, right, and we have seen it regulated, and it was like really like heavily down-regulated only in the phosphorylation level, and now we can also see it in Lisa's analysis, so which is actually really nice, right? So it's just kind of validating our findings in the cell line, yeah.
So maybe the same question is for the postmortem tissues. Do you have any effect of the ischemic effects in the postmortem tissues? Do you expect that effect as well in the proteomic data?
This is actually a really, really important question. So you always, when working with tissue, you always have the issue that phosphorylation is degrading, or actually phosphatases are getting active very, very fast. As soon as the compartments in the cell are dissolved, or as soon as any other effects occur there. So of most importance for all these phosphorylation analysis is that we have a fast fixation of the tissue, and that also the tissue chunks are not too big, because otherwise the formalin takes too long to invade into the tissue, and then the phosphorylation is rapidly degrading. So we try to control this as much as possible in this clinical setting, but of course, by taking directly clinical data from the normal pathology pipeline, this is always something that, yeah, we have to take care of, yeah.
I guess also biologically, those events somehow die out, you know. Yeah, this is a little bit unfortunate, but it requires like a little bit care, actually, when it comes to data collection.